Hi everyone, I am Dr. Shagirtha, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. In this video, we are going to study about enzyme immobilization and its applications. In this unit, we already studied about the applications of free enzymes. Now, we are going to study about industrial applications of immobilized enzymes. About studying this topic, you will understand different immobilization methods and analyze the bioconversion in immobilized reactors. First, what is enzyme immobilization? The term immobilized enzymes refers to enzymes physically confined or localized in a certain defined region of space with retention of their catalytic activities and which can be used repeatedly and continuously. We already studied about free enzymes. What is free enzymes? Enzymes are biocatalyzed. It is a protein in nature. It catalyzes the biochemical reaction that is taking place in a living organisms. So, what is the importance of immobilized enzymes and why we are going to study these immobilized enzymes? That means you are restricting a particular enzyme to a particular space. Your confined enzyme into a certain area is nothing but immobilized enzymes. Next, we have discussed about advantages of immobilized enzymes. It increases the functional efficiency of enzymes, enhance reproducibility of the process they are undertaking, reuses of enzymes, less labor input in the process, minimum reaction time and more stability of the products. Next one, applications of immobilized enzymes mainly industrial production, biomedical application, food industry, textile industry, production of biodiesel. First, we are going to discuss about industrial production. In industrial production of antibiotic beverages, amino acids are used as immobilized enzymes or whole cells. Next one, biomedical applications. Immobilized enzymes are widely used in diagnosis and treatment of many diseases like inborn metabolic disorders. Immobilization techniques are effectively used in drug delivery system, especially to oncogenic sites. Biosensor are electronic monitoring devices that make use of an enzyme specificity and the technique of the enzyme immobilization. A biosensor has been developed for detecting blood glucose in blood of diabetic patients. Next one, biodiesel production. In biodiesel has gained importance for its ability to replace fossil fuels which are likely to run out within a century. Next application is food industry. Enzymes like pectinases and cellulases immobilized on suitable carriers are successfully used in production of jam, jellies and syrup from fruits and vegetables. So let us start about physical and chemical techniques for enzyme immobilization. In this video, I try to cover adsorption, cross-linking, covalent bonding, entrapment and encapsulation. So let us talk about characteristics for a solid support. The solid support need large surface area, permeability, hydrophilic character, insolubility and high rigidity. Next, we are going to talk about the adsorption. Adsorption is the oldest and simplest methods of enzyme immobilization. Nelson and Griffin used charcoal to adsorb invertase for the first time in 1916. In this method, enzyme is adsorbed to external surface of the support. This support or carrier used may be defined different types such as mineral support, organic support and modified phosphorus and ion exchange resins. The adsorption involves weak bonds like hydrogen bond, ionic bond and van der Waals forces. Because of these weak forces, the enzyme's activity is not affected. However, changing pH, temperature and ionic strength can affect the enzyme binding. Methods of adsorption. It undergoes 4 different processes: static process, dynamic batch process, reactor loading process and electrode position process. The first one is a static process. In this process, immobilization to carry by allowing solution containing the enzyme and contact with the carrier without stirring. Second one, dynamic batch process. In this process, carrier is placed in the enzyme solution and mixed by stirring or agitation. Third one, reactor loading process. Carrier is placed in the reactor and then enzyme solution is transferred to the reactor with continuous agitation. Last one is the electrode position process. Carrier is placed near to the electrode in an enzyme bath and the current is put on. Under the electric field, the enzyme migrate and the carrier and deposit on its surfaces. Next method is covalent bonding. This is the permanent method for enzyme immobilization. This method involves the formation of covalent bonds between the chemical group of the enzyme and the chemical group of the support or carrier. Hydroxyl group and amino group of the support or enzymes form covalent bonds more easily. Carriers or support commonly used for covalent bondings are carbohydrate, example cellulose agarose, synthetic reagents, example polyacrylamide, protein carriers, example collagen and gelatin, inorganic carriers, example porous glass and silica, and finally cyanogen bromide. This is the method of covalent bonding, mainly depend on 3 types: dissociation, peptide bond, polyfunctional reagent. First one, dissociation. Bonding between the amino group of the support and tyrosyl or histolyl group of the enzyme. Next one, peptide bond. Bonding between the amino or carboxyl group of the support and that of the enzyme. Last one is polyfunctional reagent. In this method, we are going to use a bifunctional or multifunctional reagent like glutaraldehyde, which forms covalent bonds between the amino group of the support and amino group of the enzyme. Next method is entrapment. In this method, enzymes are physically entrapped inside the porous matrix. Bonds involved in stabilizing the enzyme to the matrix may be covalent or non-covalent. The matrix used will be water-soluble polymer. The form and nature of matrix varies with different enzymes. Pore size of the matrix is adjusted to prevent the loss of enzymes. Examples of commonly used matrix for entrapment are polyacrylamide gels, cellulose triacetate, agar, gelatin and alginate. So next one, methods of entrapment undergo 3 different types: inclusion in the gels, inclusion in the fibers, inclusion in microcapsules. First one, inclusion in gel. We are going to prepare a gel by using polyacrylamide gel and enzymes are trapped inside the gels. That is the first one. Second one, inclusion in fibers. Enzymes supported on fibers made of matrix material. Inclusion in microcapsules. Enzymes entrapped in microcapsule formed by the monomer mixtures such as polyamine and calcium alginate. Last method is a cross-linking. This method is also known as copolymerization. In this method of immobilization, enzymes are directly linked by covalent bonds between the various group of enzymes via polyfunctional reagents. Unlike other method, there is no matrix or supports involved in this method. Commonly used polyfunctional reagents are glutaraldehyde and diazonium salt. This technique is cheap and simple but not often used with pure enzymes. Disadvantages of enzyme immobilizations are high cost for isolation, purification and recovery of active site of enzymes. Industrial applications are very limited. Catalytic properties of some enzymes are reduced. Summary: immobilization means restricted movement of the enzyme. So it can be carried out by attaching the enzyme to a solid support. The enzyme is linked by non-covalent interaction or by covalent linkage to the solid support. The immobilized enzyme is more stable in terms of thermal stability, pH, as compared to a free enzymes. The major advantages of immobilization is repeated uses of the enzymes. Thank you.